So, Shagor, I guess you're ready. We will start with it now.
Okay, no problem.
Okay. Greetings to everybody. So, this is another of our web lecture series, and this is lecture 7, organized by the Postgraduate Department of Zoology, Vivekananda College, Thakurpukur. And I head Dr. Malavika Bhattacharjee. Welcome here today, Dr. Shagor Acharya from Department of Zoology, Vidyasagar University. Actually, I know Shagor in a different way because he is my lab mate. So he's almost just 2 years junior to me, and he was my lab junior also. So we had some lovely times during our PhD days. So how I'll move on to Shagor today. Otherwise, memories will keep flooding by. So those are some wonderful memories we had. Dr. Acharya completed his graduation from Calcutta University in zoology and got awarded in 2010 from the same university. He worked under the guidance of Professor Shopna Chaudhary, working mainly on the modulation of the proteins playing a pivotal role in cell survival and cell death cascades in glioma model. During his postdoctoral research experience, he had been the IMA Centenary Postdoctoral Fellow at Department of Biochemistry, NICET. He joined in his present capacity at Vidyasagar University on 18th April 2012 and has been serving the university since. Within his being a faculty, he has already has been the mentor of 5 scholars under his guidance. So he is the PhD supervisor of 5 scholars, and his research interests mainly include tumor immunotherapy with special reference to glioma, host-immune response of bacterial fimbrial protein, genetic polymorphism in ID among the tribal community, and molecular mechanism of bioactive components isolated from ethnomedicinal plants. Dr. Acharya has 2 UGC research projects to his credit, and during his long research career of 15 years, he has 21 peer-reviewed international and national publications, notable amongst which are his publications in Folia Microbiologica, Tumor Biology, Journal Neuro-Oncology, Cell Immunology, Asia's Pacific Journal of Cancer Prevention, Cellular Immunology, Acta Biochem, Biofix Shanghai, Anti-Cancer Drugs, International Immunology, and Cancer Investigations, amongst many. So with this very brief intro, I'll ask Shagor to start his technical session today. So Shagor, I'm stopping the sharing, okay?
Is it visible?
Yeah, it's visible. It's visible and you're audible also. A bit of a yes, but it's visible. But now your screen is not visible. That neat screen is visible, which is showing that you are presenting. Yeah, this is fine.
Okay.
Yes, fine. Perfect. Shagor, perfect.
Perfect. Okay. Thank you, Malavika. It has been a long association with you from 2004, I think, and as you mentioned, several publications of mine. I just want to mention that my first publication with your first authorship, that I started journey with my publication with you, actually, with your first authorship. I became the co-author of that journal, 2006, I think. So today I will talk about the blood-brain barrier, and especially with reference to the crosstalk between the endothelial cells, Perisites and Astrocytes. And this has been a long debated issue among the scientists, among the neurobiologists, whether these blood-brain barrier, how it will act, and what are the major role it plays. And it has been now major concern for drug delivery in brain. Now, to start with, I just want to mention that one scientist from is a very well-known neurobiologist from University of California, that Richard Damon had quoted that. It is a real naive view is that the BBB is just a wall. We, from an naive point of view, always find that it's a BBB, blood-brain barrier. Barrier means wall. But it is not that case. It is a whole series of things, not of the physical properties, biochemical properties, and transport mechanisms and metabolic barriers. So there are several barriers we'll talk about today that will actually form blood-brain barrier. Because blood-brain barrier is a concept. It's not a wall actually. You can see the wall. Uh, so before going into that, these are several barriers or blood vessels that we are found within our brain. We'll talk about much less of those things because we are only concentrated on the BBB. To begin with, that pointer. Now, when we are talking about blood-brain barrier, the blood-brain barrier mainly concentrated in the cerebro, cerebral hemisphere that we don't call it, the frontal brain. Here only you can find the blood-brain barrier in this region. Where in case of when you were talking about the ventricle, two lateral ventricle and those 1 and 2 and 3rd and 4th ventricle, the ventricles and their barrier with that of your blood that are not constituted within the blood-brain barrier or BBB, that are very leaky. So in that context, we have three barriers within our brain. One is blood-brain barrier, that is the endothelial cell that form the wall of the capillary. Second, blood CSF barrier, that is called blood cerebrospinal fluid barrier, that is the endothelial cell of the choroid plexus forming facing the cerebrospinal fluid. Fine. So that is the blood CSF barrier. And blood arachnoid barrier. We know that brain is covered with dura matter, arachnoid, and that have been lined with arachnoid membrane. And those things are basically have the barrier like this one. So in that area, you can find several layers of the arachnoid cells and tight junctions among them, and there is and CSF within that. Fine. So there is three type of barrier. One is this one that is called actually blood-brain barrier, and another one is blood and cerebrospinal fluid barrier, and next one is the arachnoid barrier. So we will concentrate among these blood-brain barrier. And why these blood-brain barrier is so much important? You know, our brain cells are very much hungry in that sense, right? Because if we talking about an brain, you find that our 20 to 25% of our entire body energy have been consumed by, or energy demand is from that of your brain. So that means, and when we compare these 20 to 25% of your energy need from your brain, and you compare the body mass of your brain, that is only considered about 2 to 3% of body mass. So 2 to 3% of your body mass required 20 to 25% of the energy. So why? Because it needs constant supply of oxygen and nutrients. So it has been two implications. That means it has huge number of ramifications among these capillaries. So it needs huge number of blood vessels. It needs huge number of blood vessels and capillaries through which can the gaseous exchanges take place. The nutrient can enter into the brain and waste products can get out of the brain. So this is the one part. And second part is it makes brain very much vulnerable, because when you eat something, when you drink something, every component coming to the blood. And if every component, the ions and the huge flux of glucose is coming to the blood, that enter into within the brain without any check, that can create an havoc. So the blood-brain barrier actually manage and separate microenvironment within the brain. So our brain is a whole set of a separate microenvironment that have been maintained by our blood-brain barrier. And secondly, it regulates the entry of nutrients. Obviously, we will see that. Second, a third, it helps the exit of the waste product, and most importantly, it controls the ions and fluids that enter within the brain and get out of the brain. And lastly, the uh, the whole CSF content, the volume of CSF content. These are also very important things. Now, before going into that, I just want to mention one or few things that we're talking about capillaries, So, what are the different type of capillaries? We have to just go through this in not in details, just give just a one or two lines about those different types of capillaries. I think you already know that thing, but for my discussion, I will just mention them. One is continuous non-fenestrated capillaries. Continuous non-fenestrated capillaries means just if I draw here, like if it is an endothelial cell lining, if it is an endothelial cell lining, within this cellular region, there is a fenestra. Fenestra means pore or window. This continuous non-fenestrated capillaries are found within the skin, lungs, and other areas that have very good tight junction among them. And next one, and importantly, these non-fenestrated and continuous capillaries have a basement membrane all along these capillaries, the abdominal surface of these capillaries. Next is the continuous fenestrated capillaries that we find in the endocrine glands, find within the villi, intestinal villi. And another one is totally different thing, that is called discontinuous capillaries, that we'll find in the liver. In that we find it will form sinusoids, blood filled. So these major gaps within these endothelial cells. Discontinuous capillaries. Now, so where is the blood-brain barrier? Where is the BBB? The BBB is in continuous non-fenestrated capillaries, but it has been evolutionary and your embryologically, developmentally, is much more tight in blood-brain barrier or capillary barrier. In that context, you can give a name of continuous non-fenestrated capillary with plus plus. Means that this slide, found cellular constituents of the in the cell, and E means astrocytes, G means function.
সাগর একটু ডিস্টার্বড হচ্ছে বলছে বোধহয়, একটু কানেকশনটা তুই আরেকবার রিপিট কর, ওই ছোট্ট জায়গাটা লাস্ট ফিউ সেকেন্ডস। Please be patient. I think so. Our speaker, due to some internet connectivity, has left the meeting. So he will be joining soon. So just be patient. I'm contacting him. Our speaker, okay. Our speaker will be rejoining back. So you just mute yourselves and wait patiently for another 2 to 3 minutes. So give him time, he'll rejoin back.
Hello?
Yeah, presentation is visible, Shagu. Yeah, its presentation is visible.
Okay,
Okay, you're audible also. Okay.
okay. Uh, mm. Um, so this is a cartoon structure of an endothelial cell lining within the blood-brain barrier. Yes. Um, so here we found that this is a luminal surface of your capillary lined by endothelial cells. These are the endothelial cells here, and discontinuous or half circle, not in full circle, half circle perisite. We'll coming to that. What is this cell? Where from this originated? This type of thing. And these are astrocytes. These are astrocyte end feet actually. These are actually brain cells. Fine. So here you find that every endothelial cells are interlinked with each other with tight junctions. Fine. And so in case of this type of, when we consider transport, we'll coming to that transport. There are three type of transport we'll find in case of these type of brain, in case of endothelial and tissue. One is called paracellular transport that actually occurs in between those the cell in the endothelial cell, in between those in the endothelial cells. So where there is no tight junction or tight junction capacity is not that much, you can find the paracellular type of transport there. And one is called transcytosis that actually occurs through those receptor mediated and movement of your ions or several nutrients. And another is the those are the lipophilic molecules directly membrane and just diffused out of the endothelial cells and coming to the tissue region. So there are the paracellular. So here in case of blood-brain barrier, this type of transport is largely inhibited because of this. Next, that is a transcytosis. That factor or transporters, and lastly, those are the lipophilic molecules that just sink into the diffuse tissue region. So that, but in case of blood-brain barrier, largely this scenario is due to the presence of tight junction, due to the presence of efflux barrier. what are those efflux pumps actually? There are 3 or 4 types of efflux pumps are there. These pumps are actually utilizing the ATP, and it uh, channel out those of the lipophilic molecules that enter into the endothelial cells through the sinking. So, the of these type of pumps, what it does, it makes the cell to get out or get rid of those lipophilic molecules that enter through the plasma membrane without any transport system. So it cannot diffuse out into the brain tissue. So these type of efflux barrier is very much important in case of blood-brain barrier. But another thing is that these are also have another implication, because most of the drugs that actually act on the central nervous system, that have been also targeted by these efflux barriers, and those drugs are, when it's coming to the endothelial cell, it just goes out of the endothelial cells by these efflux barrier. And these efflux barrier actually utilizes the ATP, because these are against the concentration gradient, because within the blood there is a huge number of concentration of those lipophilic molecules. So it actually use this concentration against the concentration gradient, whereas those transport system is actually a passive transport system, or this transport system does not uses the energy. It just takes the nutrients or any other things that within the concentration gradient or across the concentration gradient. So in the next slide, we'll see more in depth about these cellular of these type of cells. Here you can find here the electron micrograph. You can find the overlapping scenario of the endothelial cells. This is one endothelial cell and this is another endothelial cell lining. So you can find an overlap, and within the overlap you find the tight junction components. I'm coming to that one. So from this picture we find that what are the cell types that are actually within this formation of, or taking part in the formation of the blood-brain barrier. One is obviously the endothelial cell. Next one is the half-circled perisite. Next, end fit of the astrocyte. Astrocytes are the one of the major glial cells in case of brain, and one is the microglia. Not only that, actually now if we coming to the endothelial cell lining, now this picture is much more illustrious. And what are the endothelial cells here? The endothelial cell, we all know these are mesodermal derived cells, and these are modified form of epithelial cells. And you can find these endothelial cells are more or less 40 to 50% thicker than that of your muscle. So this is also an important part of formation of these barrier properties. And these endothelial cells has got highest number of mitochondria among all other endothelial cells found in our body. So these two things, I think you can understand these things, that is a highly active cell and much more thicker as well. And one important thing is that is distinctly polarized. That means this surface is called the luminal surface. Here is the blood in this area, and this is the abdominal surface. Fine. So this distinct polarization is there, and this polarization not only divides luminal and abdominal. These are also different the charge distributions among those blood-brain barriers, because here in these abdominal surface of the membrane, there is negatively charged from inside out. Fine. Secondly, I already talked about the efflux barrier. I will come into details of this efflux barrier. That actually, what it does, it actually take out the diffused lipophilic molecules too, and that coming to endothelial cell, within the endothelial cell, that diffused into the endothelial cell, goes out of the endothelial cell through these efflux barriers. One of these is P glycoprotein or MRD1, MRCD1. This is the most important one. And another important thing is that these endothelial cells lacks one of the most important things, that is called LMS. LMS means leukocyte adhesion molecules. Who we all know when inflammation, in any tissue inflammation or in diapedesis or extravasations, the leukocytes come out of the blood capillaries into the tissues. So that needs two type of adhesion. One is called rolling adhesion and one is called palm adhesion. The two type of receptors are there over the endothelial cell for those adhesion for leukocytes. So these are commonly known as, or cumulatively known as, leukocyte adhesion molecules that are actually very much absent in those cells. So endothelial cell, that in healthy brain, that's why the number of macrophages, number of there is no lymphocyte actually that infiltrating in the, or neutrophil that do not uh, come out of the blood and come into the brain itself. So tight junction, another one important thing. I have already talked about these things. Tight junction that um, greatly reduces the paracellular transport or paracellular diffusions of those nutrients and solutes, and flux of this. This is all together we can call it a paracellular flux. And very low transcytosis is also important in these endothelial cells, rather than when we compare to other peripheral endothelial cells. So next cell types is, we are talking about endothelial cells. Next is the mural cells. What is the mural cells? Within the mural cells, there you can find two type of cells. One is called the pericytes, and another one is the muscle cells, smooth muscle cells. Here you find this smooth muscle cell and this pericytes. Uh, These parasites and especially parasites have long been debated for its study because you cannot find any distinct marker for those type of cells. When you need to study one cell, you need to have identified the cells, or I have to isolate those cells. So that needs a specialized marker or distinct marker over those cell surfaces. But long it has been debated number of markers and what are the markers over this parasite. But currently few of them you can name, that is one CD13, A2, desmin, GFR3 or beta. And these are the 3-4 derived or platelet derived growth factor receptor beta that have been used for isolating and targeting or identifying these parasites. These parasites find the long cellular process. If I goes into this structure, you can find these as the parasites as long cellular processes that covers almost the capillary blood vessels or the capillary tube. Almost covers the whole capillary tubes, and most importantly, these parasites have got some contractile nature because it have got some actin protein expression. It has been found that several actin proteins have been expressed within the cell. So these smooth muscle cells and parasite actually control the diameter of the capillary, and through this the blood flow into the brain capillaries can be regulated, and these parasites take direct impulse from that of astrocyte end feet. Fine. And these astrocyte end feet or astrocytes are linked with your higher brain center, and that actually linked to this parasite, and whenever needed, the blood vessels can squeeze and blood enter into the brain can be regulated. So here another important thing, you find this two thing, that is BL1 and BL2. that is basement membrane 1 and basement membrane 2. We come into that part actually, that is what is the basement membrane in this case. Normally we found one basement membrane over which the endothelial cells were lined in other cell types. But here we can find two basement membrane. One is BL1 and BL1 is actually the inner vascular membrane or basement membrane, inner vascular membrane and outer parenchymal. Parenchymal means where there's brain tissue, major brain tissue is there, that is the parenchymal one, and the vascular one that is close to the blood vessels, fine, that's close to the endothelial cells. And where from these, now I don't know where you are aware about the basement membrane or basal laminin, I don't know, but what is the basement membrane actually is? Basement membrane is nothing but of ECM or extracellular matrix that are secreted by the cell. Fine. So it is not a, it is a non-cellular part, and we are all are aware of these extracellular matrices. So these BL1 or the inner vascular basement membrane are secreted by the neighboring cells, the endothelial cells. Sorry. Just a minute. So these BL1 is secreted by the endothelial cells and perisite. Fine. Whereas this basement membrane, that is the parenchymal basement membrane, is secreted by the astrocyte end feet. This one. So this is the responsible for secretion. Now, what are the constituents of this basement membrane? Mainly type 4 collagen. Type 4 collagen, laminin, ninhydrin, and other heparan sulfate, proteoglycan, another few glycoproteins are also there. These are major constituents. Now, if you ask me whether there are any difference between the basement membrane 1 and basement membrane 2, yes, there is a constituent difference are there. There is a different type of laminins are there, because in case of inner membrane, the inner basement membrane, that is the BL1, you can find the laminin alpha 4 and laminin alpha 5, whereas in case of BL2 you find the alpha 4 and 5 in case of BL1 and 1 and 2 laminin in case of the outer basement membrane. So the two distinct differences there. Now coming to the next part of our cells, that is the astrocytes. Where these astrocytes are come from and what these astrocytes is. So astrocytes here, we are not concerned about whole astrocytes. We are concerned about astrocyte end feet. So because whenever you see any picture here, any type of picture, you find the astrocyte end feet here, not the total whole astrocyte here. So where from these astrocytes came from? These are actually developmental process. I coming to that only. That how these developmentally these blood-brain barrier formations occur. So astrocytes is a polarized end feet. Polarized means there is a large difference between inside to outside. Inside to outside, there is a large difference in the end feet, and it contains discrete array of protein. I don't think that will be wise to discuss all those things. That is called dystoglycan dystrophin complex that are linked to your cytoskeleton. You can find those things in, there is a major dysfunction in called muscular dystrophy. In case of muscular dystrophy, these dystoglycan dystrophin mutation is there. Some proteins get mutated and those whole things disrupted and the muscle crumbling occur in this muscular dystrophy. You can find this in research. But in this case, these end feet, where this end feet structure is held by this dystoglycan dystrophin complex, part of your basement membrane by a protein called agrin. That is most important thing that here, if I can draw those things, the total dystrophin and dystoglycan complex here that are linked with the cytoskeletal structure of the astrocyte with a protein here that is called agrin, that's linked with this membrane. Another important thing I have to mention here, that these cells uh, contain an important receptor, orthogonal receptor that can call orthogonal array of particles that's called aquaporin 4. Aquaporin, what it does, it's aquaporin 4. Aquaporin, its name indicates that water influx into the, or it maintains the water homeostasis within the or central nervous system. So astrocytes is the thing that actually linked your CNS, the blood. So through astrocytes, uh, our central nervous system actually control the entry and exit of molecules, diameter of our capillary network within the cerebral hemisphere. So another important thing is the if we transplant, there is several experiments are there where actually when these endothelial cells are cultured, and you find there is basically no or very little tight junctions among them when in culture. When they are co-cultured with the astrocytes, they found that researchers found that the very good tight junction, heavy tight junction between these cells, between the endothelial cells. So that means there is an inductive effect of these astrocytes among through this induction, the endothelial cells express those genes that are actually present or actually the major components of the tight junction. There are two types of junction, tight junction and adhesion junction. Those are mainly expressed through the induction from the astrocytes. So induction we will discuss in later. Next, our cell is the last one, that is the immune cells. So there are two types of immune cells. One you can find here that have been shown here, that is a microglia. Another one is there that is called perivascular macrophages. Now, if you challenged me that where from these macrophages came, because you are already talking about that no leukocytes come through these tight junctions or parafollicular, these paracellular regions. So where from these leukocytes came from? Where from these macrophages came from? Interestingly, these macrophages are developmentally within those in the CNS. Means these, if you go through origin of these macrophages that are found within these vasculature, in they are found in these region actually. So if you look at its origin of those cells, you can find these cell lineages are from those linked to that of your that's called the yolk sac, that are directly come from those yolk sac progenitor cells. So the perivascular macrophages are two types obviously. One is called monocyte lineage and another is the this yolk sac lineage. Those are the first line of defense. Those basically function here to eat up the cellular debris, those of coming out from this type of cells. And another one is the resident macrophage of brain. That is a major one. Macrophages are very scanty, and these microglia are the major one. So it enters into the brain. These microglia enter into the brain when developmental process are still there. That is, it comes from yolk sac only. And these are the major parenchymal immune cells found within the brain. There is no lymphocyte actually within the brain. Now we come into the next part of our discussion, that this is all about the cells that constitutes your blood-brain barrier. We come into the molecules of the blood-brain barrier. Here the picture, in the right panel, you will see the picture of an tight junction and its associated proteins. So where you find this tight junction? Not in luminal surface, not in abluminal surface. These are the blood, that means here luminal surface here, and this is the parenchymal tissue, that is the abdominal surface. So none of those your tight junction protein expressed in these two surfaces. I don't know where because every protein has got its signal flow. through which the particle or the proteins have been channelized to one particular region, whether it is or called cellular transport, the protein, whether it goes into the within the your nucleus, mitochondria, or coming to the membrane. But very importantly here, the spatial distribution of this tight junction protein is very much important. It's not coming here and there. It's only coming into the apical part and the lateral membrane, apical lateral membrane of this, because if you look at here, that is the blood, that means the apical region, luminal region. The apical lateral region, you can find this type of arrangement of those of proteins. And tight junction protein are expressed, loosely expressed by all endothelial cells, but their properties, their strength, their rate of expression, and their maintenance, regulation and maintenance are largely different in case of different tissues. And it is known to have major function in formation of these blood-brain barrier, because this is the first physical barrier we will encounter here. So apart from this physical barrier, there are a few other things are there that is called the metabolic barrier. We're talking about metabolic barrier means these endothelial cells secrete several type of enzymes, especially those are peptidase and nucleosidase. Those digest mainly. Those are the small proteins, small molecules that enter into the toxins that enter into the cells. And another one is the efflux barrier. Efflux barrier means whatever comes in, it will goes out from those efflux barriers. There is a major another thing is there. So the next part of this one, the components. Those are the transmembrane components. First one, the name here, you can see, I think there's several pictures I have put here to just strongly show you. These are, I think this will make much better view for you. First one, the occludin. Occludin is a transmembrane protein, and here you can have this even number of TM. Even number of TM means whether if it come or starts from inside, it will end inside. So here the N-terminal end starting from inside, so C-terminal end ends inside. So there is 4 transmembrane region of them. So these occludin protein are major component of tight junctions, around 60 to 65 kilodalton, and the C-terminal end are linked with some adapter protein. I will come into that first. Those are called sona occludin protein. So there are major types are occludin 3, 5, and 12. When you see the arrow, it's I targeted 5. That the occludin 5 are the claudin 5. Claudin 5 are the major one that expressed within the endothelial cells of CNS endothelial cells. So not a, this is only claudin we found in the endothelial cells, in other type of endothelial cells or perivascular endothelial cells. So occludin and the claudin, there are occludin means there are 3 occludins are there. In case of claudin, there is a long debated 3 and 1, and one is major 5 and 12. And the claudin, if we just claudin are essential for this paracellular barrier formation. And if you look into the amino acid residues, you can find the domain here, and the domain is in first extracellular loop only. Fine. So domain here, in the first extracellular loop, you see the linking between 2 extracellular loops of 2 claudin proteins is very much important to formation of a physical barrier. So the most identifying and signature sequence here are found within the extra 1 within the claudin families, and tissue specific experiments we find the 25 type of claudin, but claudin 5 I have mentioned is highly expressed in the CNS endothelial cells. And then one experiment have been done that to decipher one function of a particular protein, scientists do that it actually mutate those proteins. And to actually nowadays CRISPR technologies have aided much to these decipher of these functions, and previously it is done by site-directed mutagenesis. If you can target one amino acid and mutate it and see a function of or physical manifestation of mutation. What are the implications and what are the actually physical properties of that mutation actually coming out from that thing. So that's the mice, knockout mice, basically, Claudin 5 knockout mice have seen just lack of blood-brain barrier, because that's found with full of ions and full of drugs within your mice brain, infiltrated with a huge number of leukocytes. So basically the physical barrier is lost by the only protein mutation that is called Claudin 8. So it's a major important one that's holding your blood-brain barrier or physical barrier. And occludin, on the other hand, when they mutated the occludin, you find there is basically no or very little change in the barrier properties. When scientists mutate this occludin, they found that mice developed with good physical properties of blood-brain barrier. But interestingly, they found that these brain cells are filled with calcium. So when they actually targeted one fluorescence dye with that target calcium, it finds the whole brain gets fluoresced. So find that the occludin has got a role in calcium influx, because calcium is very much important, because within the brain system, in any type of impulse coming to the end fit or into the axonal end, it opens the calcium channel. So calcium regulation within the brain, within the CNS is very much important, and these occludin mutation leads to huge number of calcium, and that mice actually died due to this huge number of calcium, and unwanted brain signaling, unwanted spark within the whole brain is occurring there. So now, next component of the discussion is the transmembrane. First was the transmembrane protein, that is the Claudin and occludin, and then these transmembrane. Another one is there called JAM. Those are called junctional adhesion complex. Junctional adhesion complex, the three types. One is called A, B, C. JAM1, JAM2, and JAM-B, JAM-A, JAM-B, and JAM-C. They're involved in the formation and maintenance of the tight junction. These two tight junction protein are linked with these adapter protein. These are known as first order adapter protein. There are two set of adapter protein is there. One is called first order, and these first order is known as ZO1 or zona occludens protein. There are three types you are seeing here, 1, 2, and 3. And another one is there that is called CASK, that is called calcium dependent serine kinase actually. So these junctional adhesion molecules and endothelial selective adhesion molecules, that is SAM, endothelial selective adhesion molecules and junctional adhesion molecules, along with the Claudin and occludin, form the tight junction. And these transmembrane protein, whereas in these, just 1 minute, coming into that, here you find immunoglobulin domain. So these JAM and these adhesion molecules, that is endothelial selective adhesion molecules, has got these IG-like domain in the extracellular region. Fine. The C terminal both those claudine and occludin and the JAMs, their C-terminal region are linked with first order adapter protein. Why these are adapter proteins? What function is adapter proteins? Because they're actually linked these transmembrane tight junction protein with that of a cytoskeleton of the endothelial cell. Fine. So the major one, first order, that is called zona occludens 1, 2, 3, and this CSK, that is calcium dependent serine protein kinase. Second order adapted protein is one is called junction and associated coil protein. I think you are heard this name called Jacob, JACOP, Jacob. That's called junctional associated coil protein. That is very important protein in case of cellular function. Jacob and Singulin, that are another one that are very much important in case of, these are the second order adapted proteins, and there are other signaling protein that are linked with these. Those signaling protein means the major one is the catenin. When this can be easily linked, you can easily link this in WT pathway. So these are those catenin actually. Those are alpha, beta, and gamma catenin. Desmoplakin and JO1 and P120. P120, it has got one name, and I forget those name of P120. But this can be linked with that of your. This is the mainly the signaling protein that can link these with. And there are other adhesion junction protein that are with that of your along with these tight junction. There is called, this is called TJ and this is called AJ. So adhesion junction and tight junction. So adhesion junction, the major one is VE, vascular endothelial cadherin. You are all aware of cadherin, integrin, flaminin. Those are things that link one cell to other, and these are called PECAM, that is platelet endothelial cell adhesion molecule. That PECAM also linked with catenin, directly linked with beta catenin. So these are few of them. The linker that is called catenin, P120, desmoplakin, that are linked cytoskeletal with that of your junctional complex. So these are all the cellular molecular components of tight junction that we have discussed here. Next part, these are all again the same thing. Not going into that details. Now coming to the another important part that is called transporter. Uh, Here you find two type of transporter here, two set of transporters. So this basement blood-brain barrier has got two distinct. I already talked about the two distinct surfaces. One is called luminal and one is abluminal. Luminal means it's facing the blood. Abluminal facing the basement membrane. So there are two type of transporter. One is called first I will talk about the known one. Those are nutrient transporter. Nutrient transporter means these are the things. That is the GALT one. You can easily link with this to the glucose transport. So these are the transporter that delivers very essential components or essential nutrients from our blood system to the parenchymal, CNS parenchymal system. The one is called GLT, and these are the nutrient. These are basically passive movement. These across the concentration gradient, because within the blood, glucose and all of this concentration is much more higher. So it easily do transport those things into the brain parenchyma. And you find their presence in, most importantly, here you can find the presence of these transporter in both the surfaces, both luminal and abdominal surfaces. Fine. So because as they are passive, that means whenever the concentration is higher, it will transport those things. Fine. So obviously, the glucose and amino acid that have been transported into the parenchyma, obviously at a higher concentration are within the blood. So by across the concentration gradient, it will transport through this direction, not through this direction, because these are passive. Fine. So GALT1 is important thing, that is glucose transporter 1, and then come the L system for large amino acid, that is called GALT1. MCT1 is also there, that are very much important for transport of several amyloid proteins. And low density lipoprotein, and these are basically these are TRF at the Trojan horses, and so these are mainly used for drug delivery. Most of the nowadays any of the drug that will deliver can be delivered within the same targeted these Trojan horses, this TFR receptors or the transporter system. The next one, the most important one that we were talking about, the next, the efflux transporter that actually form these transport barrier of the barrier function. One is the conjunction, and these are the your efflux barrier, that is MDR1. MDR1 is the PGP, that is P glycoprotein. It uses the ATP to transport small molecules. Those are basically lipophilic molecules that come
Shagor, somebody has mutated you. I mean to say, your mic has been muted. Sorry, mutated you wouldn't have been the right. So just unmute your mic once more. By mistake, I think so. Shagor, you have been. Yes. Yes. Somebody, right.
শুনতে ভালো লাগে মিউট করে দিতে, কোনো চাপ নেই।
ঠিক আছে, ওকে কন্টিনিউ কর, না আমি শুনছিলাম। ওকে, হ্যাঁ ঠিক আছে।
I think I am not boring you people. No, I will finish it up, wrap it up now.
It's not that. Some people have the habit. এটা মাঝে মাঝে সব জায়গাতেই হচ্ছে. Okay, it's not that. You continue.
Okay. Now, important is the efflux barrier. When we are talking about the barrier function, I am talking about the physical barriers that have been aided by the tight junction, and transport barriers that have been aided by these efflux barriers. What it does, it just uses the ATP and takes out those of these small lipophilic molecules that enter into the endothelial cell through the membrane, because lipophilic molecules just sink into the lipid bilayer and come into the cell, and it can sink into the abdominal surface membrane as goes into the CNS. So what it does, these type of receptor, these type of transporter, it takes those small molecule of lipophilic molecules and utilizes the energy, because it is against the concentration gradient of those things, and it puts out of this endothelial cell. So through this, it has keeping a check over these small lipophilic molecules that enter into the blood, from blood to the CNS. So MDR1, scientists, what it does is it mutates this MDR1 and form a transgenic mouse of MDR knockout mice, and it shows that increase in wide variety of small lipophilic drugs and endogenous molecules full within the CNS. So gives clear indication that these MDR1 or PGP has a role in these type of transport barriers, and when they are upregulated, these MDR1, I find that the major problems of it, that you don't find any drug that targets CNS can enter into the CNS, that any drug that enter into the these endothelial cells goes out of these large transporter systems. So now you can debate about, there is a huge debate already there about their substrate, because there is a handful of transporter in these efflux receptor, and these efflux receptors are actually taking out or transport its array of molecules. So like GALT and other transporter that has a specific substrate. So this type of PGP protein and other efflux barrier is a wide range of substrates. And even I found few studies that there it have been recorded that PGP, the two substrates don't have any analogy or structural analogy, but again they are being the substrate of the PGP. So it's a debated one, and most important one as well of in case of we're talking about the drug delivery. And now our next part will be the induction of these properties. These are the we are talking about the transport, you know. So these are the paracellular aqueous pathway that is in between two junction. So if you can put tight junction here, so these type of things will not occur. One is called transcellular lipophilic pathway, that is lipid soluble things that enter into the endothelial cell from blood, going out to those efflux barriers. There is few transporter system are there, those are specific for nutrients, those are required of this. And there are some receptor mediated transcytosis that is very low in case of endothelial cell of CNS compared to the other tissues. And another one is the absorptive transport, that is the fusion with the charge fusion. That's also very much low in case of endothelial cells, of CNS endothelial cells especially. Now here we come into the nearly end of our discussion, that is the inductive effect of these blood-brain barrier. So how the blood-brain barrier form? When we look into the blood-brain barrier, that is formed by endothelial cells. We are now knows these things. So from where these properties came from, why this barrier function is there. When we investigate that thing, we found the specialization of these endothelial cells is obviously coming from some inductive effect from astrocyte end fit. So where those induction factors are, those astrocytes secrete range of chemical agents. One of them is TGF-beta. TGF-beta all know is the differentiation. It induces differentiation of the cell. GDNF, that is glial-derived neurotrophic factors, basic fibroblast growth factor, angiopoietin-1 that have been bind with the Tie2 receptor over the endothelial cell, and these are the factors that secreted from those endothelial astrocytes, inducing or targeting the endothelial cell to become highly specialized. So other tissues, where there is no astrocyte end fit association is there, so those endothelial cells are not having this type of physical barrier property, or expression of those proteins are very much less in other tissues also. Now, whether any converse effect is there? Yes, there is. One is you find here LIF. LIF means that is leukemia inhibiting factor. So leukemia inhibiting factors are actually secreted from the endothelial cell, and it has the receptor over the astrocyte. Now, astrocytes are not within the CNS. Astrocytes are not only found in association within of your blood vessel or capillary. There are other astrocytes also that are associated with your brain cells or nerve cells. on those astrocytes, you do not find this type of specialization. So there is cell-cell crosstalk occurs within these endothelial and astrocyte end feet. Some factors secreted by these astrocyte end feet make the endothelial cell become more specialized, and the endothelial cell secretes some factors that have been received by these astrocyte end feet, and it becomes part of the barrier. That is called leukemia inhibiting factors. Now, the regulation of the regulation by the perisite, another important part is the perisite. What are the regulation is there? It's called PDGF, PDGF-BB, PDGF-R beta. These are the complex that maintain the homeostasis between the perisite and endothelial cell. These are the cell-cell crosstalk, and the crosstalk and total complex is known as PDGF-BB and PDGF-R beta that actually induces this endothelial cell, and actually the perisite induces the this amount of expression of LAM over LAM means those leukocyte adhesion factor over endothelial cells. So whenever endothelial cells grows without association of perisite, it shows the expression of LAM. So that is leukocyte adhesion factor. Fine. So there is a crosstalk between three cells actually. One is astrocyte end feet, endothelial cells, and perisite. Three junctional induction are actually very much needed for the formation of blood-brain barrier. And another important, whether any modulation of blood-brain barrier? Yes, there is some modulation of blood-brain barrier occur during inflammation that leads to huge havoc within the brain when brain edema, stroke, and this type of things will there. So these are the pathological states found where these blood-brain barrier breakdown occur, and that leads to several type of disorders. We actually, I and Madhavika, we work with brain, so we know that we are utilizing those breakage of blood-brain barriers to infiltrate our primed leukocytes to enter into the brain to engulf those cancer cells. So those are the things that are there, that it is these barrier properties is fragile. It can be broken by any time by these type of disorder, that is a stroke, trauma, inflammatory disease is a major one, where apart from the stroke and those things, the inflammation and infection as a major one, you can find these type of things. HIV infection, Alzheimer's disease is an important one where you find this aquaporin 4 that I told about, amyloid proteins accumulation there. So there is disruption in the blood-brain barrier properties. Parkinson's disease, epilepsy, brain tumors is the one I'm talking about. Pain also come from inflammation. These are the things, the pathological conditions where you find there some or less some properties of the BBB or barrier properties are being compromised. So these are the things. There is the healthy blood-brain barrier, and another one is disease. When these barrier properties is very much compromised, you find the leukocyte entered, there is lymphocyte or other neutrophil entered in the brain that are not there when there is healthy brain. So there is a huge amount of ROS generations also there when there is blood-brain barrier with the disruption. So there is an astroglial and endothelial signaling crosstalk occurs, huge signaling pathways. I am not going into that details. And each and every one is important here. That's these astrocytes, exit, and microglia. You can find these oxygen means there is a ROS generation, and prostaglandin, these are local mediators, TGF beta, and those are all the things linked with one another, and these are actually the disruptions of these blood-brain barrier. The barrier properties is very much delicate to maintain, and any instances of pathological condition disrupts these endothelial barrier can do havoc within the cells. So these are also these again showing the picture of an perivascular spaces and the membrane. It is a normal tissue, normal thing where you find no blood-brain barrier rupture, and here you find this antibody binding here and rupturing this total thing. So these are the references I used, and for a whole lot of things there. I just mentioned few of them that I'm taking those pictures from. And thank you for your listening. And if any questions is there, I will welcome this. Thank you.
Thank you, Shagor, for that illuminating talk. In fact, when I did when we were doing PhD and we talked about this brain infiltrating lymphocytes,
Yes.
it would have been helped us had you been aware of so much integrities today, the one you
Yeah.
discussed. One
Exactly.
thing is, for my personal knowledge, I just want to know from you, is there any
Mm-hmm.
permeability change that occurs at any time, making the brain more susceptible in diseased condition, making the brain more susceptible to ionic fluctuations, to the
Yes,
cells?
obviously, because those paracellular flow of flux is open in case of any type of disease condition, in pathological condition that I have mentioned. And in those cases, influx of ions is very much unregulated, you know. So in that case, it's a little bit there.
Okay, we have two chat questions, Shagor, in the chat box. One is regarding the present amount of radiations we are facing. So it's one of a PG student has asked, does
Uh-huh. Uh-huh.
radiations from the devices like headphones really make the blood-brain barrier leaky? Is this effect dependent on the amount of time or usage? Can you see the chat?
Drug.
Yeah.
I
No,
can't understand that question.
no. He's saying that whether the radiations we are constantly getting from the Bluetooth
Radiation.
devices.
Oh, the Bluetooth, radiation from those things, such as.
Bluetooth
No, I
device.
don't think this needs to be studied because we are in electromagnetic field always. There is an huge debate about things. We are now going into the 5G era, and 5G, I think that have been the radiation of the 5G is much more higher than the 4G. There's huge radiation that we will face in coming years. So yes, there is some uh, talking about this point, but I don't know, frankly, don't know about the Bluetooth radiation and the blood-brain barrier. I don't know there is any linkage of things. You can find surely.
Okay, there's another question. He's also an ex-student currently studying in BHU, Ranjit Shaw. Can you see
Mm-hmm.
the question? Yes, Ranjit's
Uh,
question.
yes, that's one. The several techniques are
There.
there to deliver the drug to the brain, like direct delivering the drug. Yes, is there into the CSF to the brain, or by blocking the Flux transporter, or by hacking the endogenous transporter system, or by yes. Every point you have written here is correct, because there is some drugs that have been designed for direct delivery. That is direct injection into the. I don't show this picture. I can show that picture here. No, right now don't have that. That are the things are there that directly injecting the drug into the CSF, or there are other parts apart from I told about that the blood-brain barrier is the not the single one that can enter into the brain. There are other arachnoid spaces are there, and other components are there through which can be targeted. So next point is the blocking of the flux transporter. Yes, that can be done, and it has been actually the combination of drugs we are using. I find that combination of drugs, that is one drug and with that actually function within the CNS, and another drug actually assisting the drug to enter into the brain. No. So one drug is blocking the flux transporter, and another drug within this blockage it can enter into the brain. Yes, that is the thing. And higher lipid solubility, no, this higher lipid solubility doesn't aid anything, because efflux barrier ultimately, efflux transporter will ultimately uh, take out of these lipophilic molecules to things.
There is another
Next,
question from same Ranjit. See, there's blood-brain barrier becomes leaky. You get that question? Yes.
yes, yes. There is neurodegenerative disorders like epilepsy, Parkinson's. How can it be leaky targeted for treat? Can the leaky blood-brain barrier targeted for treatment? Actually, no. Endothelial cells secrete those type of, or this is actually not a wall. This is actually a concept. So the whole blood-brain barrier means is interactions between astrocytes, the regulation between astrocytes, pericytes, and endothelial cells. That should be restored. And within the disease manifestation, you cannot restore those things. So you have to target the symptomatic treatment. Fine.
Uh, Tagore, are you speaking? I think so. You are not audible. Yeah, you are not audible right now. Yeah, yeah. We can hear you now again. Okay.
So anything else?
This answer, yeah. So
Stop
anybody
presenting.
else?
Stop presenting ko
Anybody else?
presenting. Stop
Stop
presenting.
presenting. So if anybody else want to ask him anything, or otherwise we'll end it. Thank you, Shagor. It was a long one and a very detailed one. It is, I think, so your
Thank you. Thank
present
you.
area of interest. Is it your present area of interest?
Right now, yes. Right now is present area of interest in terms of inflammation and yes.